Okay, thank you very much for the introduction, Philippa. So as you say, I'm going to talk about two tools that we've developed for measuring the performance of optical microscopes, PSFcheck and PyCalibrate. I'd just like to provide the acknowledgements up front in this presentation. So for the PSFcheck slides, they were developed in collaboration with Dr. Patrick Salter at the University of Oxford. And for the PyCalibrate software, the image analysis software, that was written and tested by Dr. Jeremy Metz and Dr. Michele Gentoli, who we heard from yesterday. Okay. So I'd just like to start by providing some of the motivation for this project and try and ask the question, what is the problem that these two tools are trying to solve? So when it comes to quality control in your microscopes, the status quo, as we can see at the top there, is that everyone prepares their own samples. They may be purchased, they may make them up themselves, whether they be beads in agarose, whatever it is. There's a variety of different samples that people use, different sizes of beads if they use beads. They then acquire the images themselves. Let's see if we can get the pointer. Yeah. They then acquire the images. It may be the facility manager that's acquiring the image. It might be a student. But when we've got those images, we can then put them into analysis software. It may be commercial software. It may be custom written software, various different sources of analysis. And then the challenge is to maintain the performance history over the lifetime of the microscope. So what's the vision that I have? So I suppose that the basic thesis that I'm going with here is that to get standardization, we need to go through automation. So by removing as far as possible as many different variables from this process as we can and making it as automated as we can, then we can hopefully get values that tell us about the performance of our microscope that are comparable between microscopes at a facility and between facilities. So we can get a real objective measure of the performance. And so we've made some inroads in this. We've got a standardized sample. We released a PSFcheck slide in 2019. The tougher nut to crack is the automated hardware. So this is because bracketing macros for all the different manufacturers is difficult. It's not impossible, but it's going to take a little longer. And our other contribution is through PyCalibrate, which is released this year, and that performs the image analysis, which is the same for everybody because it's all implemented through a web app. And it's not platform specific. You don't have to maintain the software version or anything like that. And then the results go to the cloud, which is freely accessible. Okay, so this is the current status that we're at. So we're using PSFcheck slides and PyCalibrate with manual image acquisition. So what are the parameters? So I'll start by talking about PSFcheck. So what are the parameters that we want to measure when we're looking to perform QC on our optical microscopes? Well, there's a list of different parameters down the left that tend to be the most popular. The PSF, we heard from Remas, for example, lateral resolution, field uniformity, so looking at possible vignetting, the absolute intensity, if we're looking at failures in our lasers, for example, color plane alignment from chromatic aberrations, scaling distortion. So basically, there's no one solution that performs all of these measures, and typically people will pick a couple of different ones. So they might use focal check beads to get the 3D PSF. The green here implies you can get a quantitative measure, the yellow a qualitative measure, and the red implies you can't use it for that measurement. So you might use as I say, a bead for the 3D PSF, and then maybe a chroma slide, for example, to get the field uniformity. And these are the most popular solutions we have. Here, the Algalite slide, for example, is able to quantitatively measure many of these parameters. But the extra parameter that we have at the end, in order to ensure that everybody can take advantage of these different standards, is cost. So this is something that for many labs, these types of standards, these slides, need to come from consumables budgets, and therefore must be kept within a reasonable cost. And that's something that we can achieve with PSFcheck, is to measure many of these parameters at reasonable cost. So which patterns do we have, and how are they used? So we have 5 different patterns. The first 2 are a 3D array of fluorescent features of different widths. So there's a smaller array where they have a 320 nanometer lateral width, and a larger one with a 650 nanometer. And these are 3-dimensional, so they're on a 10 micron pitch in X, Y, and Z. And they have a location precision of 30 nanometers in X, Y, and 100 in Z. And this can help you to identify if you've got any significant changes in distortion across your 3-dimensional field of view, if you like, if you're using scanning, if you've got any errors in your stage scanning, for the calibration of your stage, for example, that can help with that. And in terms of other parameters we can look at this large area power series. This could be used, for example, for looking at response of your sensor, or looking at the flatness of field. The grid we envisaged as being used for measuring distortion. So if you fill your field of view, and it's a large feature, 500 by 500 microns, you can measure distortion, but you can also use it to measure the field flatness, as it's such a large extent. And finally, last but not least, what we call the single point power series. So this is where we slowly tune down the feature size until we get something of sub-resolution. And these are all on a regular grid as well. And what's also quite exciting about this is that you can vary the substrate refractive index. So the conventional slide has an index of 1.57, but we can exchange this for other substrates that have indices down to 1.33 or 1.5 that we're currently developing. And this means it's very much saves a lot of time, particularly for the water index ones, for people that are developing their own slides in agarose, for example, and it can be quite time intensive. And then, of course, they dry out and you have to replace them. So anyway, okay. So we've got, they're also compatible with a wide variety of different imaging techniques. So we've taken these to labs across the UK, and we've got images from each of those. So, for example, we've got the Delta Vision OMX V3 for 3D Sim. Credit to Lothar at Oxford for being able to take these images. This is one of the larger features in the 3D array. It's also compatible with Storm, so we did this in Exeter, rather, in the group of Christian Solar, taken by Alex Clousley here. And so we're able to use this to provide some confidence on the feature sizes, the sub-resolution feature sizes within the single point power series, for example. It's compatible with multi-photon. So Ben Sherlock, an MRC fellow at Exeter, took these images using a standard Ti Sapphire laser, so for multi-photon imaging. And this is an example of that water index substrate that I mentioned before. We've used before for calibrating our own setups. And of course, it's also compatible with standard confocals, and we've used it here as an example showing the relative alignment of different color planes. Okay, and because these are three-dimensional features, we can extract the lateral and chromatic aberrations present in our system. And that's thanks to Andrew Jefferson at Oxford for that image. And so it's been quite successful. We started selling these in February 2019. We've sold over 100, largely to academia, but also some really good engagement from industry as well. And you can see from the different institutions we've sold to, and it's worldwide, and it's it's been quite well received. So we're really pleased by that. Okay. So moving on to the second aspect, the software aspect of this, PyCalibrate for image analysis and storage. So what is PyCalibrate? So it is a freely accessible web app. So if you just go to the PSFcheck website, you'll see available in one of the tabs at the top, PyCalibrate, and you'll get a page that looks something like this. If you just register, then you'll be able to log in. It's all freely accessible, and then you can start uploading your image stacks. They could be beads, or PSFcheck features, either work. As long as they're just the stack themselves, then it can perform the image processing. It all happens in the cloud through the Google Compute Engine. And then you get out at the end this nicely time-stamped calibration file. It's either available as a PDF, as a report that looks like this on the right, or you can get it as CSV files, depending on how you want to integrate it into your workflow. So the PDF gives you the file name and the metadata that's been extracted from the file you've uploaded. Any bioformats compatible file format is accepted by PyCalibrate. You get a brief overview of your field of view, showing you the features that have been identified, numbers them uniquely, so you're able to identify which feature corresponds to which in the table below. There's a table of averages from across the field of view. So obviously we're analyzing multiple features in the field of view. You can either look at the averages and the statistics, or you can look at individual features and pick out their lateral and axial extents. Okay. And so how does it work? What's the processing behind it? Well, the processing itself is not that original. It's more the implementation. And the implementation is really key here, because that provides the automation. So for a lot of freely available PSF analysis software, we need to enter different parameters into tables, so for pixel sizes or the NA of a lens, for example. And if we get those slightly wrong, then that can affect the result quite a lot, or in a subtle way that we don't expect. By using automation to extract those parameters from the file itself, it avoids these small sort of human errors. So anyway, the algorithm itself first goes through two stages. It identifies the features within the field of view using a difference of Gaussians approach. I won't go into detail here, but it's a way of being able to identify the location or the scale size of the features in the field of view. And then also thresholding provides enables us to identify uniquely different features. And once we've got a feature, we'll have a small ROI around it. And then we fit laterally a 2D Gaussian. So we're extracting a maximum, the maximum full width half maximum, and a minimum for this fitted 2D Gaussian. And axially, we also get to do a one-dimensional transect through the data set and fit a 1D Gaussian. Finally, we get to, because we fit this two-dimensional Gaussian, we also get the angle of inclination of the larger axis. Okay. I'm pleased to say I haven't been doing this all by myself, as well as the collaborators I mentioned at the start. We've had great engagement from a new initiative called Corbett, whose goal is to improve the standardization within microscopes generally. And I've been engaged with Working Group 5, who aims to define sample preparation, image acquisition, and data analysis protocols. So it's fit perfectly with our own goals here. And so it's a worldwide consortium, and the numbers, as you can see here, have been growing very rapidly since just last year. So they've been able to provide data sets for us to analyze and compare against different standards, for example. So this is just showing you how PyCalibrate performs and compares to two other freeware solutions, PSFJ and Metrology. And they've provided the 10 data sets from the same preparation protocol provided by Laurent Gelman in Basel. And we imaged across a variety of different working host confocals and processed it through the 3 software packages. And as you can see, there's not a huge amount to tell between them, both axially and laterally. You can see the bottom here, each group of 3 represents a particular data set, and the number at the bottom shows you the signal to background ratio. So this is, and for a lot of these software packages, these are quite low numbers compared to what they would usually accept. So we can see if there is any correlation, then they tend to be perform more similarly when there's a higher signal background ratio. Okay, thanks to Glenn Nelson for the analysis. And that's it, really. So there's a very whirlwind introduction to PSFcheck and PyCalibrate. We've hoped to develop an affordable calibration standard that can be compatible with a wide range of different microscopies and be able to quantify key performance criteria. If you're interested in purchasing, then go to the PSFcheck website, and we've got a screenshot here of the PDF created by PyCalibrate as well, which is also accessible through the same website. Okay, thank you very much for your attention.
Thank you very much, Alex. We have one of those slides in our lab as well, and it's been very, very useful for benchmarking our confocal microscopes. So we're rapidly running out of time for this session. So Alex, would you mind popping into the chat folder? There's a couple of inquiries coming in, and maybe you could share the link to where people can purchase the slide from. That would be really helpful.